You know, from the plastics in our kitchen to the products on our shelves, we're surrounded by a whole world of chemicals. But have you ever stopped to wonder, how do we know they're safe? How can we be sure that all these invisible substances aren't harming us at the most fundamental level, our very own DNA? Well, that is the core question we're unpacking today.
So here's our roadmap. We'll start by getting a handle on the threat to our DNA, then we'll look at the global rules for safety and check out the classic tests scientists have used for decades. After that, we're going to dive into a revolutionary new technology, see it in action in a case study, and finally explore what all this really means for the future of safety.
All right, first up, let's understand what we're actually up against. Before we can even talk about safety tests, we have to understand the threat itself, and that brings us to the science of genotoxicity.
This right here, this is the fundamental question. It's what drives the entire field of toxicology and safety science. And it's not some abstract scientific thing, right? It's a personal question that affects pretty much everything we eat, touch, and use every single day.
Okay, so the official scientific term for this threat is genotoxicity. As you can see, it's a really broad term for any kind of damage to our genetic material. A good way to think of it is anything that can break, rearrange, or create a typo in the instruction manual of our cells. And if those typos aren't fixed, they can lead to some really serious health issues, including cancer.
So with a threat this significant, how do we possibly manage it on a global scale? I mean, you can't just have every country making up its own rules, right? You need a common playbook.
And that's exactly where global cooperation comes in. What you're looking at is the logo for the IOMC, which is an international program for chemical management. One of its key players is the OECD. That's the Organization for Economic Cooperation and Development. Their whole job is to create standardized, reliable methods for safety testing, making sure that a test done in one country is trusted everywhere else.
They do this through something called test guidelines. You can basically think of these as the official, internationally agreed-upon recipes that scientists have to follow. With about 150 different methods, they really form the bedrock of regulatory safety testing all over the world, ensuring the results are consistent and comparable.
So, we've got the rule book. Now, let's take a look at the classic tools of the trade. These are the established tests that have been the workhorses of safety science for decades.
So classical tests pretty much fall into two main categories. You've got in vitro, which means in glass, where tests are done on cells in something like a petri dish. And then you have in vivo, or within the living, which involves testing in a whole organism, like a rodent. Now, there's this huge global push known as the 3 Rs. That's replace, reduce, and refine animal testing, which is why developing more powerful in vitro methods is so critical.
Now, what's really fascinating here is that no single test can catch every type of problem. It's kind of like checking a car's safety. You have to test the brakes, the airbags, and the seatbelts separately, right? Well, it's the same idea here. Scientists use what's called a test battery, a whole combination of different genotoxicity tests to look for all the different kinds of DNA damage and get a complete safety picture.
But what if we could move beyond just seeing that damage happened, and instead we could directly read the damage itself? This, my friends, is where technology is completely changing the game. This is where error-corrected next-generation sequencing, or EC-NGS, comes into play. It's a huge breakthrough that lets scientists read DNA with incredible accuracy. Let me put it this way. The old methods were like seeing smoke in the distance. You know there's a fire, but that's about it. EC-NGS is like walking right up to that fire and being able to read the scorch marks on the wall to see exactly how and where the damage started.
And the leap in precision we're talking about here is just staggering. Older methods, they'd make a mistake roughly once every 1,000 letters of DNA they read. With EC-NGS, that error rate plummets to less than 1 in 10 million. To put that into perspective, it's like being able to find a single typo in a library of over 300 books. We're talking about a whole new level of detail.
Okay, so the technology sounds amazing in theory, but how does it actually perform in the real world? Let's look at a case study where scientists really put EC-NGS to the test.
The experiment itself, as you can see, was beautifully simple. Scientists grew cells in two ways. You had the traditional flat 2D layer, and then these 3D spheroids that act more like a mini organ. For step 2, they exposed these cells to a known mutagen. That's just a chemical that causes mutations, called NDMA. And finally, in step 3, they used EC-NGS to analyze everything and see exactly what happened to the DNA.
And the results were striking. This chart shows it all.
The 3D spheroids were way more sensitive to the chemical, showing significantly more DNA damage. What this tells us is that using more biologically realistic human-based models gives us a much better prediction of what might actually happen inside the human body.
But here's the real breakthrough. EC-NGS didn't just show that there was damage, it identified the chemical's mutational signature. So think of our DNA code as being written with 4 letters, A, T, C, and G. Well, EC-NGS was able to pinpoint the exact typo that NDMA causes, swapping one letter combination for another. It's like finding a chemical's unique fingerprint right at the scene of the crime. The study's authors wrapped it up perfectly, concluding that this approach enables a full range of genotoxicity endpoints to be monitored, making it a potentially powerful human-based platform for genotoxicity testing. It is a huge step forward for creating safer, more human-relevant science.
So we have this incredibly powerful new technology. But what does it all mean? For us? For the products we use every day? And for the very future of safety itself?
Okay, let's recap the key takeaways. First off, we get more human-relevant results by using these advanced cell models that better mimic our own biology. Second, this powerful new in vitro platform helps us move away from animal testing, which is a huge deal. And third, the unprecedented accuracy of EC-NGS gives us a depth of understanding that we've simply never had before.
And this all brings us to a really fascinating final question. As our technology gets powerful enough to find even the tiniest needle in the haystack, the smallest possible change to our DNA, how does that change our definition of what's safe? In a world where we can measure risk with such incredible precision, how do we decide which risks are actually acceptable?